right. Hello, everybody. So my name is Nick Berendricht. I am a postdoc working with Arit Peleg in the computer science department. I will be kickstarting off some talks from a few people in our group, talking about research that we are interested in getting some help in. So I'm going to kind of just dive right in and give a little bit of an introduction to what our group does, which is modeling complex systems, specifically bio-inspired complex systems. And so you may wonder why, reasonable question. And really the reason is that these systems are pretty ubiquitous in nature, whether you're talking about herding animals, schools of fish, or different groups of even human individuals. These are dynamics that exist all around us, and something really fascinating about kind of complex systems is the emergence of collective behavior. What individuals can't accomplish alone, they can come together as groups and achieve great things. And so we're really interested in trying to understand the fundamental ways in which these systems arise, self-organize, and then act as a group of these individuals, all with their own individual preferences, but working towards larger goals. And so our lab kind of focuses on two different organisms in order to study these complex systems. The first being honeybees, which Danielle will talk about after me. But what Noel and I do is we study fireflies. So this is actually a picture that we got from our field season this past summer in the Great Smoky Mountains National Park. And you can see all these fireflies in this long exposure photo where we were doing a lot of experiments. And so you may ask, okay, that's kind of maybe a strange organism to pick. Why are you doing that? And there's several different reasons why studying fireflies is particularly a good way to tackle some of these questions that we're thinking about. So first, just like a little bit of background on fireflies in order to motivate that. So there are many different species of firefly around the world, and each of those species has a unique pattern that they will flash and send a signal that they use for mate selection. And so what you can see kind of in this picture here on the right is a bunch of, in this case, male fireflies kind of flying over a trail and doing their characteristic flash, which in this case is kind of one flash every half second or so. So you can kind of see the trail of them as they move along here. And one of the really nice things about these flash patterns is that it's an on-off flash signal. So we can approximate them as just a binary string. And that's a lot easier than, for example, if you were studying human communication, it's a very complex multimodal form of communication via vocalization. It's harder to kind of decode what's going on there. Whereas with flash patterns, binary strings, we have a pretty good handle on that. And something that's really fascinating about some species of firefly is that if there's a high enough density of them, then they will synchronize their flash patterns. So they'll actually, all collectively time, out their flashes so that there's this collective beat going on across, maybe sometimes hundreds of fireflies. You can, so for example, you could see it in this picture. If this was actually a movie playing out, you would see kind of waves of light propagating along this trail in a synchronized beat. And so this actually in itself is a very interesting question because we have some hypotheses as to why they might synchronize, why might that be advantageous for the group, but we don't know entirely, all the specifics, and we don't know why some species synchronize and some don't. But one thing that we are pretty sure about is that all of these flashes, they're communicating information across this kind of large swarm of fireflies. And so that is something that we can more directly study. So kind of the thing that I'm really interested in and that I'm talking about in this project that I'd be interested in having some help on is specifically how things like spatial perception, visual occlusions, and kind of the layout of the environment that fireflies live in affects the flashing structure that arises as part of this synchrony, and also the flow of information through that network. So you might see here in this picture on the left, all these different little dots or fireflies in another long exposure photo. Some of them are kind of in different parts of this trail. Some of them are hidden behind brush, and yet they're still all synchronized together, even when maybe they can't directly see every single firefly in the collective. So how does that process happen? And so can we actually then measure how information flows from one end of the swarm to the other, for example? And then this is kind of getting at this larger question of is this a benefit of synchronization? Does synchronization really help information flow more easily? And is that something that we can put a quantitative figure towards? So I want to talk to you about this project that we're working on that we're calling sort of the info transfer project. So this is data that we just collected this past summer, and I want to describe a little bit what the data looks like, what we did in order to get it, and then where we're looking to have some help come in. So this was all collected in the Great Smoky Mountains National Park this past summer. And we did a lot of experiments where we captured individual fireflies. We placed them in small acrylic boxes, and then we placed them inside of tents like this one on the left, covered in tarps to prevent any external light pollution. And we also had GoPro 360 cameras set up in these tents so that we could record everything that was going on inside the tent over a period of, let's say, 10 minutes. And the reason we put them all in these individual boxes is that constrained every firefly's movement so that we could track an individual's position over the entire duration of that recording. So, like I said, at high densities, fireflies will naturally synchronize. But one thing we wanted to really study is, do these effects persist even at low population densities? And so my experiments, we had not only fireflies in the tent, we also had an LED in the tent. So that's kind of something that I alluded to maybe a little bit here. You can see that one of the other advantages of working with fireflies is that we can communicate in a way with them via LEDs. They respond to an LED light. If you play the same pattern that is typical of that species, they will interact with that light. And so what we did is set up an LED in the tents that is programmed to the species-specific flash pattern. And then we put fireflies in the tent to try and measure how much they are able to synchronize with this LED source. And in order to study these sorts of kind of environmental layouts and visual occlusions I had talked about, we've actually placed barriers. So here, this black line being one of these barriers. And we had some fireflies that we call conduits that are able to directly see the LED and synchronize with it. But then some other fireflies that we call targets that can't directly see the LED. So the only way they could synchronize with it is via this conduit of information flow. And so that's what the experiment looks like. And what we get out of this data, and I'll leave kind of how we get this data to Nolan to talk about a little bit in his slides. We're able to pull out of the movie the position of every single flash that happens in every frame of that movie. And then from that, we can extract an actual time series of activity. So here on the left is that position graph that I've shown, where each of these kind of three clusters that you see is either a firefly or an LED. And on the right is this here. So the blue is the LED. The red is this conduit that is able to see the LED. And then the green is this target that is unable to see the LED, but can see the conduit firefly. And then x-axis here is representing time in minutes. And so what we can see is that actually this looks promising, that fireflies are able to entrain to the flashes of the LED, even if they cannot directly observe what's going on. And so this gives us a really rich data set because we have many different configurations of this experiment to really precisely see how different spatial topologies influence information flow throughout a swarm. So what are we looking for help with? First, what we're interested is actually working on a much more efficient way to take this sort of raw data that we have on the left and transition it into labeled data that's here on the right, using different clustering methods, which there are several that I've listed here, but that's an open project to explore. And once we have all this labeled data, we can go in and we want kind of a very traditional network analysis approach to this. You could do some edge weight analysis in order to really infer the strength between every agent in the tent, and then look and see dynamically, how does that change over time? Does the target firefly take longer to synchronize with the LED than the conduit, which can directly see the LED, for example, would be kind of a first order question to answer. So these are really the things that we're interested in getting help with from somebody like you. on this project. So I'm actually at this point going to move into questions and make sure that my other lab mates have time to give their talks. So if you're interested, we'll have all this contact information at the end of our talks as well. But you can email me. My email here is nicholas.barendrick@colorado.edu. And if you're interested in working on this sort of thing, send me either a CV or resume and just some times that you would want to meet in order to discuss this project. So I think I'll take questions for maybe, 2 minutes, 3 minutes, and we'll have some time at the end as well. So thank you for listening. And feel free, you can just unmute yourselves and ask questions.
So did you also test with maybe like multiple LED setup and see which one it synchronizes with?
So that's a very good question. So we did not do it with multiple LEDs, but we do have also multiple conduits. So multiple fireflies that can see the LED, one firefly that cannot see the LED, but can see all of those multiple conduits. So we don't have multiple LEDs in this particular experiment. So that's a good question. Thank you.
Hello.
Yes.
Thank you very much. So the question I have is, the LED light that is being used for this experiment, does it have to have the same color the fireflies are emitting, or can you use a different color for this experiment?
Yes, that's a great question. So the short answer is yes, they do need to have a fairly specific color. So you can maybe see it if I go back to this original picture. But most of these flashes look yellow, but there's a few little streaks here that look more green. You can kind of see where my mouse is. And it's actually a different species, and its lantern that it flashes with produces a slightly different color. So you can see it very nicely in this sort of picture. So we do need to choose the color of the LED to match the color of the firefly because their visual perception is very much centered around the wavelength of light that corresponds to their species flash color. Luckily for us, the yellow that is used by the species is a very commonly available LED, so we did make sure to pick one, but it wasn't too difficult to do.
All right, thank you.
Yeah, welcome. All right, I think I will stop there and let our next speaker take over, but we'll have time for some questions at the end.
All righty. Sweet. So let's go ahead and talk about more fireflies. So my name is Nolan Bonney. I'm a third-year PhD student in Uri Peleg's lab. And today I've got a couple of different firefly research projects that we're looking to bring on maybe 1 or 2 master's students, maybe even an undergraduate student as well, to just assist with the research with. At the bottom is my email, which will also be at the end of the presentation. But let's go ahead and just talk about a quick overview here. So first thing we're going to talk about is just like the briefest of introductions to bioluminescent communication. And then we're going to talk about our first project, which mainly centers around infrared experimentations. We're going to talk about the motivations of why we're even doing that before getting into what we would like to do in the future, which involves some deep neural network models and just for identifying fireflies in infrared video. And then we'll get into our second project, which centers around the species of fireflies that tends to flash at sunset. And I'm going to talk all about how our flash detection algorithms and process works and what that looks like. And then finally, we'll talk about how, we want to maybe expand that flash detection pipeline. So again, to be clear, there are 2 different distinct projects here. Both of them we're fielding interest in for some extra research help. And great. So let's talk first about bioluminescent communication. So fireflies are one example of a species that use bioluminescent communication within a swarm. So fireflies have the ability, they have 2 lanterns on their abdomen, and they have the capability of pulling in oxygen to their lanterns, and then that produces a chemical reaction, which creates the very bright flash that we're all familiar with. previous literature and a lot of work in our lab has centered around the temporal aspects of these flashes. So specifically, we're looking at these binary flashes over time, right? And how maybe that information moves through networks, the mating dialogue that happens between fireflies. We've looked at how the flashes relate to time in a lot of detail. But something we're very interested in looking at is how fireflies utilize spatial movements along with their flashes. So a lot of different other bioluminescent species have very interesting movement patterns when they flash. For example, there's a species of fish that right after it flashes, it makes a sharp turn in a direction, probably because it's some evolutionary defensive mechanism. When you flash, you're also advertising your location. The main reason that we haven't done this yet is that it's dark outside and fireflies are very small, and it's not quite possible to see fireflies fly around in the dark. Um, and something that, people ask is like, oh, like, why can't you just light up the surrounding area outside? Well, so fireflies communicate with light-based communication methods. Specifically, they're utilizing the visual spectrum around 500 to 700 nanometers. And so visible light is not an option for illuminating the background, but what is an option is sort of peeling outside of their visible spectrum into the infrared side of this. And that's sort of what started our infrared project here. So the basic setup that you're seeing in the background here is we have two 360 GoPro cameras that can see in infrared. And we have lamps on all sides and even above the tent, flooding the tent with infrared light, and we release fireflies into the tent. The idea is that we can track them as they're flying around. And so we have a lot of questions that we would like to ask about the data. Specifically, how are fireflies moving when they're not flashing? Because there really hasn't actually been any literature done into that question, because it's very hard to even get into a place where you have the data to answer that question. Second question, maybe as we talked about before, have fireflies maybe evolved or adapted some sort of movement-based evasion, maybe like the fish that flash and then turn right away. Maybe there's something there. And broadly speaking, I'm very interested in how information can be encoded in movement. So we have certain species that actually move while they're flashing too. And there's a lot of evidence in bioluminescent communication through different species that movement can be an attribute of information encoding, but we haven't been able to answer those questions yet for fireflies. So here's sort of the motivation. This is, what we would like to eventually answer with this data. But before we can answer these questions, we need to process the data. Oh yeah, of course. And so here is just, again, the basic experimental setup. We have an IR lamp which shine onto the tent wall. So the tent walls get really illuminated with infrared light, so that when fireflies fly between the tent wall that's illuminated with the IR light and the 360 GoPro camera, we can see the shadows that they make by blocking some of the light from the tent wall. And so here is the top-down view where you can see the two GoPro cameras in the middle, and they see in every direction. The idea of two cameras is that we can then triangulate the positions of fireflies when we track them through both cameras. And right on the end here, on the right side, we have two panels that sort of demonstrate, this is real footage that we've collected, but it demonstrates the challenge where in panel B, we can see a fairly clear definition of a firefly. And if you're thinking this isn't a clear definition, I understand that, but that's definitely a firefly. And with our eyes, when we just watch the video, it's really easy for humans to track the firefly in every case. But in panel C, you can see an example of where a computer might get confused. Uh, Infrared ecological data is notoriously noisy. So not only are the infrared lamps not always shining a very consistent amount of light, but our cameras have a lot of grain to them because they're trying to film in the infrared spectrum. And we're out in the nature, so we have tents and there's wind and the wind likes to ripple the tents and it creates all sorts of different background noise, things that might confuse traditional computer vision techniques from actually identifying firefly from noise. But so this is the challenge that we're facing, and this is where we would ideally like a master's student, an undergraduate student to come in and take on this project. Is, we've sort of exhausted the traditional and advanced computer vision methods that aren't quite accurate enough to handle all of the situations that we're looking to cover. And so that's sort of what you're seeing at the bottom of the screen. The left photo, that's just a binary image of what we're seeing after background subtraction. So there's a firefly, that main blob there. And then on the right, that's an example of just a multi-frame stack of a firefly being tracked across frames. So, we have run traditional CV methods, but they're not accurate enough. So we're looking for someone to build a training data set of fireflies versus not fireflies, implement a deep neural network model to locate and track the fireflies throughout the entire video. And then ideally, the dream here is a little bit of model validation, process automation. So really, it's the complete package. If you're looking to increase your computer vision skills, if you're looking for a good CV project to put on your resume, this sort of encompasses not only a very difficult problem, but a complete problem that you wouldn't be skipping many steps here. You would get to do it all. And so, yeah, so this is project one, infrared tracking of fireflies. And now we'll talk briefly about project number two, which centers around this particular species of fireflies called Photinus pyralis. And I sort of consider them a sunset firefly because Photinus pyralis only flashes at sunset. And we have a whole bunch of questions that we're really interested in answering about this very unique species. Mainly, do they synchronize in high densities? How is information transferred throughout the swarm? Because remember that when a firefly flashes, that's its communication to the swarm. And then finally, and most importantly, can we even track their flashes reliably? So over the last summer, I collected a lot of data of this species, but sunset fireflies present a lot of very unique and interesting challenges that might make flash detection a little bit more difficult. And so before we get into the specific problem here, let's talk generally about how flash detection works and what we have done in the past to get flashes from our videos. So essentially we have, hour, 2 hour long videos and fireflies are flashing in them. And we want to find all of the firefly flashes, find the location. And so here's an example of a real raw image from a video. And you can pretty much make out that every yellow dot here, that's a different firefly. And you can tell that there's varying intensity of the flash. There's varying sizes of the flash. There's a whole bunch of reasons. Maybe the firefly is looking away. Maybe the abdomen is not pointed towards the camera. There's a whole bunch of ecological reasons that might prevent us from getting, uniform flashes across the frame. But you can also see, like, up in the top right, that's going to be some like moonlight coming through the canopy. You can see some trees in the background. But so this is our raw image. And our first step is to actually just grab the green channel from this. So all images are RGB, red, green, blue. We just grab the green channel because as we've talked about before, fireflies can actually only see in more or less this green channel, and their flashes are also in the yellow to green zone. And so once we grab the green channel, we have a background subtraction model that will take out sort of the noise here. So if you see the difference, you can sort of see the trees and the sky. And then once we take out the background, it's a very clear what is the foreground here. And then we simply just binarize the image, and every single white dot you see here is a Firefly flash. And so this is our standard way of analyzing these videos. The problem that we're facing with Photinus pyralis fireflies is that since they flash at night, or since they flash at sunset, there's a lot more visible light in each frame. And that visible light is going to be across all sorts of different color spectrum. And, you know, our flash detection algorithm has really only been written for and implemented on nighttime videos. Photinus pyralis, they flash during sunset, which means there's a lot more noise in each frame. You might be able to see the sun and the reflections on the water and reflections on blades of grass and wind wrestling the trees in the background, which creates a very noisy environment that's not conducive to like simple background subtraction. And moreover, what I think is also kind of difficult is that the lighting conditions are constantly changing in the video, that between the start of the video and the end of the video, we've gone through the entire sunset. So we start very bright and we end very dark. And so on the next slide, you can see an example of this, right? The bottom left is a frame from the start of our video where you can actually see a Firefly flash. Is that one little guy in yellow right in the middle. And the same exact area and spot, I took a same, you know, screenshot from the same video, but just at the end of the video. And you can see how different it gets. You can still see that one little dot right there. That's the Firefly too. So we want to be able to capture all of the Firefly flashes from every circumstance, right? In these different dynamic lighting conditions. So we're looking to bring on someone to expand our flash detection algorithm to handle some of these different conditions, to handle finding fireflies in bright conditions, and also within the same video, be able to find fireflies in dark conditions. And perhaps most difficultly is, you know, once we can verify, or once we can detect those fireflies, how do we verify the model that we're using to detect flashes? You know, how do we evaluate our flash detection performance? All of these things are very important. This is a very unique and challenging problem within computer vision, and could offer a lot of unique, not only CV skills, but also like data analysis skills and model evaluation skills. Yeah, and so these are my two projects. We have the infrared tracking, we have the sunset fireflies. My email is at the bottom. Please do not hesitate to reach out. I love talking about this stuff. I love thinking about fireflies. And super happy about the prospect of getting to work with some of you. And if there's any PhD interest in general, I put Arit's email at the bottom as well, but you're also more than welcome to email me about that too. Sweet. And so I think we have a few minutes for questions, if we have any.
Hello, hi. I would like to know, is there a specific kind of light you use, like the watts or the wattage or the lumens? Is there a specific kind of light, the LED lights you use for this project?
For the infrared project?
Yes, please.
Yeah, so I mean, I'm certain that our lamps have specific wattage, but that's not at all important to the experiment. The whole point of the infrared lamps are just to illuminate the tent and to fill it with as much infrared light as, you know, kind of as possible, so that we can see fireflies when they fly between like the tent wall and the camera.
Okay, thank you.
Of course. Okay, we got a question in chat. Does your current model track both motion and light or just blinking light? Current model. So I'm guessing you're talking about the infrared. Currently, infrared is just tracking motion at the moment, but we will need it to track motion and flashing. But I mean, if we solve the motion problem, it's very easy to extend to like a flash detection. That's probably just like a very simple, maybe even like an AlexNet deep neural network on top, just to classify flash versus non-flashing characters. So we're currently just doing motion. That's the current focus, but both would be great. And we have another question in chat. Have you tried any image segmentation algorithms to remove background for detecting Firefly flashes at sunset? So our current flash detection algorithm does do some very, very, very basic background subtraction, but it wasn't really built for the purposes of detecting Firefly flashes at like sunset, and specifically at the site that we recorded Photinus pyralis at, there is like a running river going through, and the sun is setting straight into the cameras, and it's reflecting off the river as well. And if you can imagine, those conditions are always changing, and the sun is a sort of yellow that kind of gets picked up as Firefly-like sometimes. So yeah, there's a whole lot of questions and a whole lot of different algorithms that we could try to use, but I think that is the project, is to try different algorithms, try different methods, see what does work. I see there's a hands up. You can.
Yeah.
Yeah.
Hi. So I had a question that for the blurring that's occurring in the infrared radiation, the movement tracking and at the
Yeah.
end where the blurring is occurring, have you tried Gaussian filtering for that?
We definitely have. And the
Okay.
the basic idea here is that, yeah, like we essentially tried every standard computer vision technique in the book, even some of the more like cutting edge within the last 2-3 years, things that have the models that have come out there. And
Okay.
it just hasn't produced any results that are as accurate as we need them to be.
All right. Yeah. Thank you.
Sweet. So I'm going to go ahead and wrap up questions just so Danielle can have enough time. But please, if there are any more questions, please feel free to reach out. Super happy to answer any questions you may have, and hopefully we can connect and interview some of you for these research positions. So with that, I'll hand off to Danielle.
Hi, everyone. I'm Danielle. I'm a postdoc in Orit Peleg's group, and I'm working with honeybees, and specifically honeybee swarms and how they adapt to changes in their environment. So honeybee swarms, in nature, there's tens of thousands of bees, and you might see them hanging from a tree branch or other surfaces, and they'll stay there for several hours to several days while they're looking for a new hive. And during that time, they will experience many environmental fluctuations, but one is temperature, as you know, it cools down at night and then heats up during the day. And when the ambient temperature is changing, it was observed that the swarm morphology is also changing. So that at cold temperatures, you can see this more compact swarm, and then you can see it sort of expanding in warmer temperatures. And it has been hypothesized for a very long time that the internal structure looks something like this in different conditions. So in cold conditions, the swarm is more compact, and it's hypothesized that there's sort of a dense outer layer and then a looser structure inside, while in warm temperatures, maybe there's some channels that allow heat to leave the swarm. And so our questions are kind of in these varying conditions, cold weather, warm weather. How is the structure of the swarm changing? And how are those changes made? And then why does the swarm modify its structure? And so we study this in the lab so we can control the temperature very precisely. And we build our swarms under a board. So in this yellow cage is a queen, and bees go to the queen and form this kind of hanging structure. And we put this swarm into an X-ray machine. We control the temperature, and then take images, essentially doing a CT scan to get a 3D reconstruction of our swarm. So
one pass that really looks. Oh, okay. I remember one.
Oh, sorry. Okay, so that sort of 3D reconstructions are what we're looking at. And then here in the bottom, we're looking at just a cross section through that 3D swarm. So this is kind of what the inside looks like of a swarm that might look like this on the outside. And the white is our bees and black is sort of empty space. So you can kind of see that in warm temperatures, the swarm is less dense. There's more open space inside the swarm. It's also sort of larger compared to cold temperatures where, we have much higher density. Bees are closer together, and the swarm is also smaller. So if we just zoom into some local region in the swarm, we can look at the orientation of individual bees and notice that in warm temperatures, these bees are sort of more disordered compared to cold temperatures where we see that they're highly aligned in many areas. And this is sort of what we're interested in thinking more about. How do individual bees look inside the swarm? So we kind of can see like the bulk structure changes between between different ambient conditions, but how are individuals sort of contributing to those changes? And so to look at individual bees, we have a neural network to sort of, I should say, a very preliminary neural network to take our 3D input data and identify whether every voxel, is a bee or not a bee, and then get a segmented swarm that looks something like this, where there's like 6,000 bees in this swarm. And now we can kind of consider that every bee has sort of like its individual identity. We can look at the orientation of those bees, how that's changing in time, how close a bee is to its neighbors. And with this kind of data, we can answer a few questions. So the first is still, can we identify individual bees in our reconstructions? And can we do it at different temperatures and for different regions in the swarm? And then once we have something where every bee is, an individual, can we ask questions like what the network structure looks like under various conditions? How does that structure change when you go from cold to warm temperatures? And then how are bees modifying the local structure to generate these global morphological changes? And so bees are connected by like holding other bees' legs. So to rearrange, they either need to change the length of these connections or they need to detach and reattach. And so how are bees able to make those large changes where we saw the swarm like shrinking and expanding? And yeah, these are some of the questions that we're interested in. And now we can open it up to questions about any of the projects. And then if you have other questions later, you can get our contact by email. And you'll find more projects at peleglab.com.